Hello, everybody. My name is Jackson Parrish, and I am a recently graduated master's student from ASU. I graduated with my master's in biology, and I was a part of a wonderful research lab called the Brain Epigenetics and Altered States Research Lab, otherwise known as the Bear Lab. Now, before I get started, I just wanted to say a warm thank you to PsychedelX for inviting me to participate in this talk conference. I am very excited to talk about my research and kind of my journey and how I got here. And I'm looking forward to the future of where this goes. So, yeah, without further ado, this is Psychedelics, the gut microbiome, and aging. But before I get into it, I want to talk about myself for a little bit. So, my story in the psychedelic field starts out in 2022. This is Baby Jackson. Now, as you can tell, a couple of things have changed since then. I've grown a little bit. My weight, my hair, and a couple of other things that I won't get too in depth about. But Baby Jackson had a great idea on a whim, and that was to change his major from chemical engineering to microbiology. This was because he had just watched Fantastic Fungi, the Paul Stamets film, if you are not familiar, and he was interested in fungi. He wasn't sure how to start, but the closest thing that ASU had to offer was microbiology. So he decided to bring the fungi education to ASU and started a student club called the Shroom Society at ASU. Now, in the Shroom Society, we went foraging. We had guest speakers come out and talk about how to grow your own mushrooms. We created grow kits with edible mushrooms, of course, uh, under the approval of Arizona State University. And we had some really good times. And yeah, believe it or not, mushrooms do actually grow in Arizona, just up in the forest in Flagstaff. So, flash forward 1 year. I had the life changing experience of a study abroad. I went to New Zealand, and oh boy, my heart, a piece of my heart is left in New Zealand. It is so beautiful. Kiwis are the funniest people, and they're the best people too. And there's lots of mountains and really good coffee, like feeds your soul type of coffee. Delicious. Do you know what else is in New Zealand? Lots of mushrooms. So I don't know if you guys recognize any of these mushrooms. Maybe the one on the middle left you might recognize. Won't go in depth about what it is. Maybe later in the presentation. But the one on the right is the Were Were Kokako, and this is a blue mushroom that is native to New Zealand. It only grows in New Zealand, and they actually have it on their $50 bill. They have this blue mushroom. So uh, this was a part of the reason that I love New Zealand so much is it's a giant rainforest island essentially, but it's temperate, and so there's lots of mushrooms there. I would look down and there was mushrooms left and right. Another thing I loved about New Zealand was I took a class called microbiomes, and in this class, I we discussed about different types of microbiomes. So the overall definition of what a microbiome is a community of microorganisms that resides in a given environment. Now, this includes bacteria, fungi, viruses, and even archaea. Now, if you're confused about what archaea is, look on the right. This picture is a thermal pool. This is one that's in New Zealand, of course. And uh, they're extremophiles. Archaea are generally extremophiles, so they live in extreme conditions. They're kind of like bacteria, but just in extreme conditions. And so essentially, there are communities of microorganisms in many different environments, including this. But I think that the type of microbiome that most of us are more familiar with, or at least have heard of, is the gut microbiome. So, what is the gut microbiome? The gut microbiome is a community of microorganisms that resides in our gastrointestinal tract. The gut microbiome influences many things, specifically metabolism, immune function, and even our neurobiology. Uh, our gut microbiome helps synthesize neurotransmitters, which can change how we show up in our behaviors. This happens through the gut-brain axis, which is a bidirectional. Think of it as a two-lane highway that goes each way. The brain sends signals to the gut, and the gut sends signals to the brain. And this communication is mediated through or controlled by neural signals, immune signals, and even through metabolic pathways. So, where do psychedelics come into play here? Well, in that class, microbiomes, while I was in New Zealand, I got assigned a paper to present in front of the class. And I'm very glad that this was the paper I was assigned because it was about ketamine and how ketamine interacts with the gut microbiome in rats. Now, for those of you who don't know, ketamine is a dissociative anesthetic with some psychedelic properties that in low doses it acts as an antidepressant, but in high doses it's an anesthetic. So it does not have antidepressant effects when it is used as an anesthetic, but it does not have those same anesthetic effects when used as an antidepressant, right? So uh, something very interesting is that in low doses in mice or rats, it it changed the gut microbiome in a beneficial way. But in the high doses, it did not, which was it boggled my mind. I was like, what is going on? Why is it changing the gut microbiome in beneficial ways in low doses and antidepressant doses, but not in anesthetic doses? So that led me to lead to wait, but what about other psychedelics? So I searched up psilocybin and microbiome to look up if there was any research on it. And there was this a proposal article where it was an overview article proposing that psilocybin might psilocybin's effects could have something to do with the microbiome. So as soon as I saw this article, I was like, I got to look to see if ASU has any research going on about psychedelics. And luckily enough, there was a lab, and that's the Bear Lab. So in 2023, this is still while I was in New Zealand, I sent a Hail Mary email to the Bear Lab, the Brain Epigenetics and Altered States Consciousness Research Lab. Uh, they were doing a study in which they were giving psilocybin to aged mice. And so I proposed, hey, I'm a microbiology student who's interested in microbiomes. Can we take poop samples from these mice that are being given psilocybin and look at the gut microbiome to see if it changed? So uh, my hypothesis was that the psilocybin treated aged mice were going to exhibit an increased gut microbiome diversity compared to controls. So one professor who was not in the lab but was tied to the lab, and I quote, told me, "This will not work. The gut microbiome is shitty." Uh, I agree. The gut microbiome is shitty because it's fecal samples, but it did work. So just an overview of where we're at so far and like how my story kind of came this way. I had an observation that ketamine had an impact in the gut microbiome at low doses, but not at high doses. Additionally, I looked at other psychedelics like LSD and psychedelic like compounds such as MDMA. And there was one study with psilocybin, but they were all inconsistent and they were all done with young mice. So I asked a question: what impact, if any, does psilocybin have on the gut microbiome, but in aging populations? So I had an action, and that was to partner with the Bear Lab at ASU to collect poop samples or fecal samples of aged mice that were given psilocybin and compare the microbiomes to mice that were given saline, which is the placebo. So, in terms of how the study worked, the mice were given two injections of psilocybin 1 week apart, or saline 1 week apart. After the two injections, they were given 22 days of behavioral testing, and then we collected their fecal samples and sacrificed them. Now, we used an aging model. So these were 10 to 12-month old C57 mice, which are the normal lab mice that people use. Now, if we look on the right, we can see that the middle-aged mice are kind of in that 10 to 14 month range. The ones that we've used, they were 10 to 12 months old. So that's 38 to like 43-ish. Now, this is before mice start to die at higher frequencies, but it is right around the beginning of whenever age-related biological changes and behavioral changes start to occur that we see in mice. So, additionally, these mice were co-housed with mice of the same sex, so only males live together and only females live together, as well as if one mouse in a cage was given psilocybin, all of the mice in that cage were given psilocybin. Now, why is diversity important? I've mentioned this word a couple of times. So, what does that have to do with anything? So, in terms of diversity, we're usually referring to the alpha diversity. Now, the alpha diversity is characterized by the diversity within a community. There are several different types of ways to measure the diversity, but in terms of the aging gut, the alpha diversity is one of the best measures that scientists can use. So, generally, when we see age increase, we see the diversity decrease. So, if we look at this chart on the right, we can see that there's two different terms, the richness and the evenness. Now, the richness is the number of different species that occur. So, if we look at these charts, we can see sample one has 5 different types of bacteria, so it has a 5 richness. We can see sample two has 5 different types of bacteria, so it has a 5 richness. But sample three only has 3 types of bacteria, so it has a richness of 3. So generally, when we see an increase in richness, that is a good thing, generally. Evenness, that is another term that's important. Whenever the species are present in similar abundances, that's pretty even. So if I give you 10 M&Ms and I give you 10 different colors, that means that there's one of each color. Therefore, that's a very even sample. But if I give you 10 M&Ms and I give you nine nine purple M&Ms and one green M&M, that's not a very even sample, right? So that's the term evenness. Now, these two things are used in statistics to describe the alpha diversity. So a reduction in microbial diversity is seen in aging guts, and this contributes to systemic inflammation, cognitive decline, and metabolic dysfunction. So, what did we observe in the mice that we gave psilocybin? One, we saw an increase in the alpha diversity, which is probably a good thing. We saw a decrease in the most abundant type of bacteria in their samples, also probably a good thing. It was a reduction of firmicutes. And the observation three was a sex specific observation. We saw a reduction in the ratio between firmicutes to Bacteria but only in the male mice. Now, there's 2 metrics of alpha diversity that we use, and that's Shannon diversity and Simpson diversity. Uh, they're essentially both looking at the same thing. But Shannon diversity looks at the richness and the evenness, and the Simpson diversity looks at what is the probability if you select 2 bacteria that they are going to belong to the same type of bacteria. So, Shannon diversity, we saw an increase in Shannon diversity, as we can see on the right. Let me move my face. There we go. We can see that in terms of the saline treated mice and the psilocybin treated mice, we saw an increase in the psilocybin treated mice of Shannon diversity. And same with Simpson diversity, which is really cool. Additionally, we saw a decrease in the most abundant type of bacteria, which is also probably a good thing. We saw a reduction in firmicutes and a marginal increase in Bacteria. Third observation is that sex differences continue to be uncovered in psilocybin and psychedelic research. We saw a reduction in the firmicutes to Bacteria ratio, but only in the male mice. So this ratio is very important in aging, as well as depression and anxiety. In aging populations, as well as depressed and anxious populations, Generally, we see an increase in this ratio. However, in the male mice, we saw a decrease in this ratio, which is mind boggling. Well, not really, but it's cool. So, in terms of comparing our results to prior literature, we saw an increase in the Shannon and Simpson alpha diversity index, but prior studies have shown inconsistent results. This is the first study that has showed any evidence that psilocybin increases gut microbiome diversity in any model, which suggests that age is a large factor. Additionally, we saw an altered microbial composition, a decrease in firmicutes, and an increase in bacteriodota. This is consistent with prior findings, as certain firmicutes bacteria, such as the lactobacillus species, we see sensitivity to psilocybin exposure. In terms of how to interpret this, we can interpret this as psilocybin induces restructuring of microbial communities in aged mice. Why should I care? I mean, these were middle-aged mice. What does this have to do with people, right? Well, I'm going to go back to mice to answer your question. Psilocybin extends lifespan in aged mice and decreases aging-related biomarkers. So I have two pictures in here that are important. The first is on the left. It's the graph. And as we can see, there's two lines in this. The black is the saline, that's not psilocybin, and then the green is the psilocybin. So we can see that the psilocybin treated mice, the survival of the psilocybin treated mice was higher than the not psilocybin treated mice. And if you remember and recall back to that graph earlier, mice start to die around this time. So the fact that the psilocybin treated mice lived longer, there's some biological things that are going on that we don't fully understand. Now, if we look just at the mice, between 18 months and 28 months, they were on psilocybin for 10 months, once a month. And as we can see, their fur grew back. Like they look healthier, which is really interesting. Now, I'm not telling you that your grandma should go and take psilocybin, but we're going to get to a point, maybe, where your grandma might uh, maybe should be taking psychedelics. So, if you're interested in learning about the biological effects, here is a chart that I have made. Take a screenshot of this. I'm not going to go into it at all, but I made this for my thesis. Take a screenshot of this if you're interested in learning more about the studies. Reach out to me. I'm happy to connect you with any of those studies. So, if we're interpreting on a bigger picture, like why should I care? Psychedelics seem to influence multiple aging related systems, such as neural systems. We see an increase in neuroplasticity. We see changes in immune functioning. We see changes in metabolic functioning. And now we see beneficial changes to microbial systems in the gut microbiome. These observed microbiome changes that we see are aligned with the known systemic effects of psilocybin. And in conclusion, psilocybin acts as a systems level modulator of aging processes. In terms of future directions that I'd like to see, uh, in order for us to give psilocybin to your grandma or your grandpa, we need to do this research in humans too. I'd like to see research like investigating that. Additionally, I am curious if the microbial changes that we see are they drivers or are they effects of the systemic effects of psilocybin? Additionally, I'd like to see sex specific differences investigated further in the treatment with psychedelics, as we have seen in many studies that there are sex related differences that happen with psychedelics. In terms of my acknowledgments, I'd like to acknowledge all the funding partners: Arizona Alzheimer's Disease Research Consortium, Barrett the Honors College, TGen Helios Scholars, the Brain Epigenetics and Altered States Research Lab. Thank you so much, as well as the mice and the professors from the lab, and of course, PsychedelX. Thank you so much. Reach out to me if you have any questions. I'm always happy to talk through. Thank you.